Accounts of Alzheimer's disease are usually cast as chains of causality. We look for an early distal disruption, perhaps peripheral inflammation or a shift in gut ecology, and trace it forward through a sequence of intermediate steps until it eventually reaches the cortex and destroys a synapse. But if we reverse this approach and look at the terminus of the disease, a precise molecular convergence emerges. 5 independent laboratories, working across different disciplines, have traced the structural execution of Alzheimer's synaptic failure to a single physical location. They all point to a specific protein called cofilin 1, governed at a single residue, serine 3. This matrix shows the published findings from these laboratories in human Alzheimer's tissue, and they present a glaring contradiction. On one side, researchers demonstrate that amyloid pathology drives the disease by adding the phosphate to serine 3. On the other side, independent groups demonstrate amyloid drives synaptic failure by removing the phosphate. The field largely ignores this conflict, dismissing it as measuring different disease stages or cellular compartments. But both directions claim to be the primary structural driver. They cannot both be monotonic. By leaving this directional contradiction unresolved, we obscure the actual mechanical failure. The evidence dictates that our basic assumption about how Alzheimer's pathology operates at the synapse is incorrect. We assume a disease must have a single pathological direction of travel, that a protein is either too active or too inactive. The true signature of Alzheimer's synaptic failure is a loss of governed range, not a simple displacement of a chemical mean. Testing bulk tissue homogenates or synaptosome fractions mathematically blinds researchers to this reality. A bulk sample takes millions of individual synapses operating in different microenvironments with different local receptor densities and averages them into a single number. If half your synapses are pinned extremely high and the other half are pinned extremely low, your bulk average will look perfectly normal, or it will skew slightly, depending on which population you sampled more heavily. To resolve the contradiction between these 5 laboratories, we have to abandon bulk averages entirely. Instead, to find the truth, we must look closely at the non-monotonic physics of the actin cytoskeleton itself. The dendritic spine is a dynamic volume of actin filaments, rapidly polymerizing at one end and fracturing at the other in a continuous cycle. The structure persists only because this rate of turnover is maintained. Cofilin 1 is the master switch that controls this actin treadmilling. When it is active, it binds to the filaments and alters their structure, governed strictly at the serine 3 residue. This graph maps cofilin activity against structural stability. At a low ratio of cofilin to actin, sparse decoration introduces stress, severing the filament. But at high ratios, the molecules switch, stabilizing the network and bundling actin into pathological rods that strangle the neurite. Because this switch reverses its own function, pushing the residue in either direction from the healthy center results in structural pathology. There is no direction of travel from the baseline that is safe. A healthy neuron relies on this dangerous mechanism. To change its synaptic strength and store a memory, the cell must execute a brief, controlled event called an excursion. During an excursion, calcium enters the spine, triggering a sudden drop in serine 3 phosphorylation to activate cofilin. The network severs, allowing the spine to expand. To prevent destruction, the protein renin arrives, driving phosphorylation back up to stabilize the new, larger volume. Structural memory is an active, funded capacity to leave a set point on instruction and safely return to it. Governing this precise molecular excursion requires biological resources. It is highly fragile. Managing this requires a perfectly placed signal, like renin, and a massive metabolic ATP reserve for actin turnover. The cell must also guarantee the return of surface receptors and maintain a shielded baseline free of chronic ligands. If any of these 4 physical preconditions fail, the neuron loses its governed range. The excursion cannot be completed safely, and the actin balance collapses. A layer V pyramidal neuron in the human cortex is born before the individual is. It is never replaced. It carries its cumulative structural burden for up to 90 years. Holding thousands of spines requires constant actin treadmilling. Over 8 decades, this obligatory maintenance consumes the cell's energetic reserve. The metabolic budget is exhausted simply holding the structure in place, leaving nothing to fund an excursion. Simultaneously, the endosomal recycling machinery degrades. Receptors that should be returning to the surface to read incoming stop signals are stranded inside the cell. The cell also carries an exposure load. The condensed perineuronal net that shields the healthy neuron slowly dissolves with age, leaving the membrane undefended. The immediate consequence of this degradation is that the unshielded receptors are continuously flooded by chronic ligands present in the aging brain, specifically ambient amyloid beta oligomers and the complement fragment C4d. The cells that die first in Alzheimer's disease are those carrying the highest geometric, electrical, and metabolic requirements. Their selective vulnerability is the arithmetic consequence of carrying an immense structural load until their biological budget goes bankrupt. This bankrupt biological budget settles entirely at the serine 3 switch on the dendritic spine. Stripped of its metabolic reserve and its protective matrix, the cell can no longer afford to initiate the cut, and it lacks the receptors to terminate it, even if it could. The chronic flood of ambient ligands converts cofilin from a transient informational signal into a permanent, uninformative setting. The residue is held at a constant value rather than driven to it. Because receptor densities vary from synapse to synapse, the microgeography of the failing cortex is split. One synapse, overwhelmed by rho kinase tone, is pinned in high phosphorylation and stabilized to death. A neighboring synapse, overwhelmed by amyloid at a different receptor, is pinned in dephosphorylation and severed into transport-blocking rods. This extreme synapse-to-synapse variability explains the contradiction between the 5 laboratories. When you homogenize a tissue sample, your bulk measurement simply reports which of these 2 pathological extremes was more abundant in your specific test tube. This statistical distribution compares the state of millions of synapses. In a healthy brain, the distribution of phosphorylation looks like a tight bell curve clustered around a functioning central mean. The Alzheimer state warps this data. The curve flattens and splits into a highly separated bimodal distribution. The mathematical mean of this diseased bimodal graph sits in the dead center of the graph. It is exactly the same mean as the healthy bell curve, hiding the dysfunction entirely from bulk assays. Applying monotonic drugs to this bimodal state, like ROCK inhibitors designed to push phosphorylation in only one direction, is mathematically dangerous. If you push the entire curve toward dephosphorylation, you relieve the synapses pinned high, but you simultaneously accelerate the destruction of the synapses already pinned low, driving them further into rod formation and neurite collapse. Treating a loss of variance with a one-directional intervention guarantees harm to a vast subset of synapses. This framework offers a cheap, falsifiable prediction. True Alzheimer's pathology must be measured by synapse-to-synapse variance. That variance will be elevated relative to age-matched controls, even in tissue samples where the mean appears perfectly normal. If this holds, drug developers must stop targeting downstream kinases and phosphatases directly. You cannot cure a broken steering wheel by permanently locking it to the right. The correct therapeutic target is the restoration of the neuron's biological budget. By repairing endosomal recycling and matrix shielding, we can return dynamic range to the cofilin switch. Alzheimer's is not a novel pathological process that arrives to attack a cell. It is the progressive loss of a load-bearing cell's ability to govern its own biology, exhausted after a lifetime of structural maintenance.